Okay, so here we go. Good morning and a very warm welcome to this innovation workshop as part of On Helix 2023. So on the first day of the digital days in the lead up to the in-person day on the 6th of July at Baber Research Conference. My name is Alicia Gallier, Business Development Manager at One Nucleus, and it's a pleasure to have you on the call this morning. Speakers and the delegates have joined. So this session is actually an innovation showcase. So what that means is that the companies presenting are going to give insights into how they are helping to and their role in enabling translation. So we have a selection of companies to present to you this morning. Some are in the earlier stages, startups, a couple as part of the Accelerate cohort at Baber, which is very exciting as that's also where One Nucleus is headquartered, and also one of the finalists for our BioNews Round Award as well. So I'm really excited to hear the company's presentations and what exciting science they're helping to develop. So if you're not presenting, just a quick note to stay muted with cameras off. The chat box is enabled, so you are very welcome, and we encourage questions throughout the session. And that's my job, just to keep an eye on that chat box and feed that to the speakers. I've allocated 15 minutes per presentation. If you think of a question outside of that, don't worry, just pop it in and we can pick it up towards the end. There's no problem there. And then also you do have access to the conference app as you're part of the session here. So you can also use that to pose any questions to the speakers who are also on the app, should it come to you post the session. So with that, I think I've gone through all the housekeeping there, very brief. I'm going to kick off then by inviting Frederick Claesen, who is the co-founder at Gigabiome, to share his screen and then to take us into the first presentation. Over to you, Frederick.
Brilliant. Thanks a lot for the introduction, Alicia. Can you guys see this?
Yes, we can. Thank you.
100%. Thanks a lot for inviting us to give a short overview of the company that we recently founded. So we are a really early stage startup, Gigabiome, and we develop precision diagnostics for antimicrobial resistance using the wonderful world of the microbiome. So let me just get this going. So imagine a patient that are being admitted to hospital, showing some potential signs of infection, and upon first examination by a clinician based on sort of empirical knowledge and some rough guidelines, this patient is given a course of antibiotics. At the same time, this the clinician requests for some culture to be done on this patient, and then after a couple of days, this antibiotic sensitivity report comes back, showing a bit more detail around the resistance profile of this patient, and then another course of antibiotics is then prescribed to this patient. After some time, this patient might get better, is then discharged from hospital, giving another big bag of antimicrobials to be taken home to prevent some further downstream infection. And then after some time, this patient might then get sick again, then consult potentially a local GP, which then prescribes another round of antibiotics to this patient. So this cycle of continuous intake of antibiotics is very common in patients that suffer from chronic diseases. So these chronic diseases include things like chronic liver disease, which we have a major focus on, as you'll see, but also chronic kidney disease, different respiratory diseases, just to name a few. What ultimately happens to these patients, this delays their optimal treatment. They are continuously suffering from infection, and then this ultimately then causes them, especially in the case of liver cirrhosis, is a high cause of mortality within these patients in chronic diseases. So what we try to do at Gigabiome is to develop a precision diagnostics to prescribe the right antimicrobial at the right time in an accurate, personalized, and quicker manner. So our founding team consists of myself, Professor Rajiv Jalan, and Dr. Saeed Shawi. Rajiv is a consultant hepatologist at the Royal Free Hospital and has a lot of expertise in 20, 30 years of expertise in chronic liver disease, especially in patients suffering from liver cirrhosis. And Dr. Saeed Shawi is a senior lecturer at King's College London in Systems Synthetic Biology. And I'm also a research associate at King's College London. So we've worked together for approximately 5 years now, made through consortium programs such as MicroPredict and CarbAlive, which these consortium programs all aims to address different parts of liver disease and the microbiome and how the microbiome impacts the development of chronic liver disease. So what do we actually do? So we've, over the course of the last couple of years, we've built some state-of-the-art profiling software for characterizing the microbiome at both gene and species level. So we currently, our pipelines currently can take oral samples as well as gut samples. What puts this apart from sort of other well-known pipelines in literature is that we functionally characterize these communities. So what we then do is this functional characterization, in this case, in terms of antimicrobial resistance. We can then relate this back to which species are actually driving this antimicrobial resistance, and then ultimately associate this back to clinical features. Using this pipelines, we've then developed some large pan-metagenomic resources. So this is an example of our gut atlas, which we call it. So this is across 19 countries and 23 different diseases almost. We then sort of do this functional characterization around how this sort of transient change of the microbiome composition on both a species and again a functional level. This is an example of how we did use this in antimicrobial resistance. So because we have this large cohort of data, we then, for example, looked at how antibiotic resistance in the microbiome differs in healthy and cirrhotic patients, and then we can then see which genes are responsible and which microbes are responsible for driving this resistance in these large cohorts of patients. What we do at the moment and what we are developing over the course of the next year or two is predictive models to be able to use these microbial signatures, both genes and species, as well as clinical features, to use this for building AI models for prediction. So this is once again a really simple example where we've in this case actually used only these AMR genes within the microbiome to be able to distinguish between resistant and non-resistant individuals with quite all right accuracy. And once again, this is one of the really early on models that we've built only using antimicrobial resistant genes. Our business strategy is to partner with hospitals that have sequencing facilities available and to provide a diagnostic software platform. So how we envision this especially in terms of liver cirrhosis patients, is when this patient is admitted to hospital, is to provide them with a antimicrobial resistance card, as we term it, and then which gives an accurate report of resistance within this patient, and then making a recommendation on which antimicrobials are the best to prescribe to this patient at that particular point in time. In 2023, the antimicrobial resistance diagnostic market was valued at about $4.1 billion, with an annual growth rate of about 6.2%. But what is really exciting is we've seen this really influx in the human microbiome markets and very high, almost exponential growth rate predicted over the next 10 to 15 years of about 25% up until 2023. So the global rise in antimicrobial resistance, as we know, is becoming a massive problem across the globe, and this created a market need for real rapid and accurate diagnostic tests. But at the same time, this advancement in sequencing technologies, which are driving the human microbiome market, creates a sort of perfect alignment, putting Gigabiome, we think, right at the center to address this very, very complex issue. So a bit of our vision and where we are going with Gigabiome. So as Alicia mentioned, we are currently part of the Accelerate at Babraham cohort of this year. We are raising some pre-seed funding at the moment for the development of our, specifically our liver disease platform, GigaL. This funds, we want to use this funds for sample collection and marker validation, which we hope sort of around at the end of the year, this will give us a more solid proof of concept to open a seed funding round within 2024 to expand this platform to other chronic diseases. So because we have a real focus on liver disease and a lot of expertise, but we ultimately want to focus this on a more broader chronic disease problem. And then obviously to use this to increase our data science and lab team. In the long run, we also envision this to this diagnostic platform to also inform a therapeutics platform, because we know that the microbiome is very transient and changes because of a lot of environmental factors. We do think that a lot more data will enable us to be able to more and with more precision being able to target resistance within this community ultimately before infection. But this is something that I'm happy to discuss again in the questions. So thanks a lot. That's a short introduction to what we do, and like I said, I'll be happy to answer any questions that you might have, or have a chat afterwards. Yeah.
Thank you very much indeed, Frederick. That was really interesting. If anyone has any questions, please don't be shy. Feel free to drop those into the chat box. Just something that came to mind, you know, we're on this session, we want to be sort of collaborative and sharing. If there's anybody else joining at the moment who is also at a similar stage in terms of pre-seed, what tips, what kind of learnings could you offer for sort of this stage of growth? Because there's many challenges, isn't there?
Yeah, I should have included that as a question.
Nope, just yeah. Anything you can to you, Frederick, if to anyone who is also at a similar stage to you, what tips could you offer?
At this point in time, so being so early on, I think a good tip from our side would be, I think it's very useful. The accelerator programs around London and Cambridge is a good tip, I would say, to try and apply for as much of them as possible, because there is a lot to manage so early on. And that's been really helpful for us to develop that our ideas further and to solidify that. So that's a good tip, and find that community, I think is really useful. Being a first-time founder, especially, that community is really important. Although you have some I have some co-founders around me who's done this before as well in a couple of companies. It's still important, I think, to find that community that really helps solidifying that. Yeah, I think that's really important.
Thank you. And aside, you said you're going to move the focus eventually from the liver cirrhosis. What do you have any ideas of indications that are sort of top of the list to go to next? I don't know, cancers.
Yeah, so I think there's a lot to do in different stages of liver disease as well. It's something which we are, like I said, really interested in. In terms of other chronic diseases, we have some collaboration with people with chronic kidney disease as well, which is potentially the next stage after that. Yeah.
And do you need what sort of stakeholders are you looking for involvement from then? What sort of groups would help you propel forward the most then?
To propel forward the most at the moment, I think sequencing partners, as well as people with more at this point in time, I guess, access to samples, right? I think it's the brilliance of AI is still very, very much dependent on as much data as possible. So I think that is the key here, right? So I mean, and especially in terms of resistance data, we've been struggling quite a bit to get good public data, because I mean, even in terms of resistance, we struggle to build even models with just like simple classifier resistant versus non-resistant, just because this information is not available most of the time from public repositories, right? So and then further sort of clinical data gets even more difficult. Yeah. So I think that's the biggest hurdle at the moment in terms of partners is data and sequencing. Yeah.
Thank you for highlighting that. Any thoughts, comments from the audience? You could just unmute if it's easier than using the chat box. No? I won't we'll move on, but like I said, you know, it's fluid. You can put in questions for different presentations at different times. Don't worry about that. So
yeah, I'll pop my email in the
thank you,
chat as well.
Frederick. Yeah, thank you very much indeed for sharing that update on Gigabiome. So moving on then, I'm just going to add, I'd like to welcome Steve Myatt to the virtual floor. So Steve's going to talk to us about Macomics, and you're happy to share your slide deck, aren't you, Steve? So over to you. Sorry,
Absolutely.
the CEO of Macomics. Over to you, Steve.
Absolutely. Yeah, thanks for the introduction. I'm not going to be able to see the time when I'm presenting, so keep me to time, Alicia.
I'll keep an eye on that.
Yeah, if I start going on for a little bit too long, just let me know. Give me a heads up. I'll speed through the slides.
No
yeah, so
problem.
it's really a pleasure. Thanks for the invite to speak today, and it's really a pleasure to introduce Macomics to you all. I have a presentation that will give you a little bit of background to the field, and also talk you through kind of some of the key highlights of what we're currently doing at the company and kind of where we're going next. So can everybody see this okay? I'm going to put it on full screen mode.
I can see the slides. Yeah.
Yeah, okay.
Just waiting for that to go to full screen.
Are we there?
Not from my side. Have you gone to, is it either the icon, I think, in the bottom right, the little, like, looks like a TV screen, I think.
Yeah, I did press
or
on
under
that.
slideshow at the top, is there a full screen option?
Normally if you just you do that, it should be fine. This should normally present the screen now, but I'm not that familiar with Zoom. No look.
No, it's still in
Oh.
Do you want me
Okay.
to have a go, or
no, I'll tell you what I'll do. I'll minimize the tab at the top. I'll collapse that to the side, and then that's more or less full
fine.
screen.
Yeah.
Okay? Yeah.
Thank you.
That's good. That's probably good enough for the purpose of the
Thanks,
presentation,
Steve.
as long as everyone can see. Okay, yeah. So no, it's a pleasure to present today. I'm Steve Myatt. I'm CEO of Macomics. I joined the company about 2.5 years ago now. And my background's in originally as a pharmacologist, and then I've worked in sort of commercialization and exploitation of early stage intellectual property for the last sort of 15+ years now, in a variety of roles in tech transfer, venture capital, and biotech. So this is an overview of Macomics. We're a macrophage therapeutics company. We're focused on oncology 100% right now. We have a focus on novel biology. So we're approaching first-in-class targets, and also first-in-mechanism approaches against validated targets. We were founded by Jeff Pollard and Luca Cassetta 3 years ago now. We're really global leaders in macrophage immunology. We've also hired a great team who I'm going to introduce in a few slides' time. So we're not just a portfolio company, we're also a technology company. We've built an in-house macrophage drug discovery platform, at the heart of which is new technology that allows us to gene edit human macrophages. And that's really significant and really enabling for us. And I'll give you some examples of that in the coming slides as well. So we've had some major corporate events as well in the last sort of 3 years, and actually some very recently. So we've raised, it's actually over $20 million now, that number should be. since the company was formed 3 years ago. And we're going to be raising another financing sort of towards the end of this year, early next year. And we've also done our first corporate partnership. So in March this year, we entered into a collaboration and partnership agreement with Omo Pharma, a Japanese pharmaceutical company that many of you will be familiar with, for an undisclosed discovery program that's in the immuno-oncology space. And behind that, we've built our pipeline. That's the overall shape of the company. This is our current pipeline. So our lead program is a first-in-mechanism LILRB1/2 dual antibody therapeutic, and this is currently in IND studies, and the next real major milestone for the company will be that program progressing into first-in-human trials. And we expect that to happen next year in a number of different solid tumor oncology settings. We also have the program that's partnered with Ono. This is currently at discovery stage, and we're building out our pipeline behind that as well. So we're targeting effector cell communication, how macrophages relate to other cell types in the immune system. And also we're leveraging our discovery platform to identify new therapeutic targets as well across a number of different disease settings. So that's the overall shape of the company, but I'm going to sort of take a little bit of a step back and talk you through some of the background to macrophages, then kind of a little bit of data on the lead program and the pipeline. So what are macrophages, and kind of why are we interested in them? Well, they're a type of immune cell. As I'm sure many of you know, they're part of the innate immune system. So they're a completely separate biological and orthogonal regulator the immunoregulatory state inside tumors to T cells. But obviously T cell therapies really have changed the landscape in terms of how patients are treated over the last 10 years or more than 10 years now in some indications. But there's still some really major challenges. So although immunotherapy has been effective in some patients, the majority of patients that are eligible for immunotherapy still don't receive a benefit. And what we're really interested in Macomics is new and distinct mechanisms for modulating that tumor microenvironment that doesn't rely on direct targeting of T cells, which can be confounded by T cell exhaustion or anergy, and for which there's already a number of therapies in either launched or many in development, but targeting macrophages. And why? One of the reasons we're interested in macrophages, they're actually the most abundant immune infiltrate in almost all type of solid tumors. So if you were to take a section through a patient's tumor and stain from a macrophage marker, you would see macrophages within almost every different sort of tumor microenvironment context within that tumor. So both at the periphery, in the necrotic core, in the sort of invasive margins of that tumor, you would find different types of macrophages potentially across almost all types of solid tumor. And macrophage infiltration tends to correlate with poor patient outcomes as well. And we actually know that's true across almost all modalities now. So if you look at chemotherapy or targeted therapy or immunotherapy, if you find immunosuppressive macrophages in them tumors, it tends to predict a worse outcome for that patient. And on the left here, you can see an example of that using this sort of nomenclature that's quite common in the field, but it's actually quite outdated now, where an M1 is like an anti-tumoral macrophage, a pro-inflammatory macrophage, and patients that have higher levels of those tend to do better. But we also know that the state of a macrophage prior to therapy impacts the outcome. So they're not kind of a passenger or a cell type that's sort of passive in that microenvironment. They're actually determining how patients respond to therapy. And we know this, for example, for checkpoint therapies in breast cancer, where if you have high levels of immunosuppressive macrophages pre-PD-1 therapy treatment, you're less likely to do well on therapy. But we also know that successful immunotherapy can actually actively reprogram macrophages as well inside both patients and in mouse models as well. So it's quite a complicated picture, but overall the key message is that there's a lot of these cells inside tumors, and they tend to predict poor response to therapy as a result of being an immunosuppressive cell type in that context. And we think they're very clinically, as a result, very clinically relevant cell populations. And they're also a little bit distinct to T cells in the sense that there's no such thing as a macrophage cold tumor. Almost all solid tumor types have macrophage infiltration, different types of macrophages, different portions of the tumor maybe, but it's almost universal in most types of solid tumor. And there's specific biomarkers now which allow us to understand which are the particularly negatively prognostic macrophage populations and also which predict poor outcome to therapy. So they're actually really clinically relevant. They're there, they're abundant, they predict patient outcomes, and they're determining how patients respond to therapy. Now, what was the sort of unique insight that led to the formation of Macomics? It was really our founders sort of in a piece of work that started in New York when they worked together at Einstein 15 years ago, Jeff Pollard and Nuku Cassetta, and they brought it back to the University of Edinburgh when the lab relocated, and continued that work. And that led to really a really breakthrough insight. It was the first example in the field of what we now call disease-specific macrophage reprogramming, or tissue-specific reprogramming. And basically, in a nutshell, what this means is, for a very long time, people had known that if you compare a macrophage in a normal sort of tissue context to a macrophage in a disease context for the same tissue type, for example, breast cancer, the macrophage in normal breast tissue would look very different to the macrophage in the breast tumor. But what had been not actually understood or appreciated until this point in time was, if you compare macrophages between two different disease settings, the way in which that disease changes that macrophage also varies quite significantly. So if you compare endometrial cancer and breast cancer, for example, the way in which those macrophages change in those disease settings is actually, there's actually almost no overlap. That's a really extreme example. Most disease sort of comparisons aren't like that, but that's a really extreme example of how actually the reprogramming is very disease specific. So the company was founded on basically that insight and human sequencing data across multiple different disease contexts, where we could mine that data set to try and understand those changes and use that understanding to think about how we might rationally target those macrophages. And that's what we've done. So we've built something we call the Macrophage Atlas. So we have over 40 data sets from 7 different tissue types, from normal and diseased or cancer tissue. And we've built and really confirmed our founders' initial insight that there's a lot of heterogeneity in macrophages, and that they tend to be, to some extent, perturbed on a disease-specific way. And what we've been really interested in, how then we use this from a therapeutic perspective. We're not a sort of data science company, fundamentally, we're a therapeutics company. And in conjunction with that, we've seen some really exciting developments in the field more broadly. So we've now seen the first data sets coming out in a phase 1 context, where therapies targeting phagocytosis in certain blood cancers are actually being really profoundly active as monotherapies, really deep and durable responses in some settings. And we're also seeing the first reprogramming data sets, where people are trying to convert macrophages from like a tolerogenic state to a pro-inflammatory state, generating positive early clinical data. But there's still a very long way to go, and we still don't really understand how to maximize that therapeutic signal as a field in clinic. That's something we're having to predict based on preclinical models currently. So how do we go about that in Macomics? What's our kind of strategy? Sort of build on that, early data set and our insight into disease, the disease sort of specific changes in macrophage biology. Well, I think the first thing is, as a field, we just have to completely move away from this idea that there's basically 2 macrophage states, diseased and non-diseased, and that we can effectively really simply categorize them in that way and drug them on that axis. So what we're doing at Macomics is we're identifying disease-specific macrophage populations where we see that comparison between normal and diseased tissue. We're looking for shared and tissue-specific immunoregulatory pathways. So we're obviously really interested in what we would consider to be kind of dominant immunoregulatory pathways that are shared across multiple disease settings, but we're also interested in really niche-specific contexts where we understand exactly which patient population would benefit, and where we're targeting biology that's sort of almost private to that particular niche. And then we're thinking about that from really new sort of ways of targeting macrophages to targeting their role in tumor immune escape, functioning as molecular sensors in the tumor microenvironment, and communicating with effector cells. And the overall objective of all of these different approaches, it basically boils down to the same thing. The aim is to activate the immune system in the tumor more broadly than just the macrophage. So to really try and address that, we built this discovery platform. We call it Enigmac, and it combines 3 things: the Macrophage Atlas that I've already sort of introduced, coupled to more relevant functional assays. One of the really big problems in this field is that the in vitro assays have just been really poor. So they're using macrophages that really don't represent human macrophages. They're kind of immortalized. They're derived from cancer cell lines often. They're not immunosuppressed. The genes that are expressed in a patient aren't found in these systems. So we've sort of tackled that problem head on to build more relevant functional assays using a stem cell model that allows us to differentiate those stem cells into human macrophages that are fully dependent on all the growth factors they should be. They're fully plastic, and they can display very highly immunosuppressive behaviors if they're sort of differentiated in the right way. So we've developed next generation kind of in vitro assays, and then we bring those 2 things together. We bring the human data sets together with functional assays through our gene editing platform. And this has really been a big leap forward for us. It's taken us sort of through the last 3 years to really get this to the stage it's at now, but we could now, with very high efficiency, genome edit these fully human fully representative macrophage models. And we can do this at the stem cells stage, and we can do this at scale in the macrophage stage as well. So for the first time, actually, as a field now, because gene editing macrophages, I mean, gene editing is quite straightforward nowadays in any different context, and people use this as the toolbox to target discovery, target validation all the time, as you'll know. But this has actually been a massive problem in the macrophage space. So it's not easy for many reasons. And by kind of addressing that problem, we can now, for the first time, go from human gene expression data sets through to actual targets that matter at a functional level. So understand all that heterogeneity, all that complexity, to pick the right targets by being able to gene edit those macrophages, and as a result, do all sorts of target validation, target discovery sort of work in our more functional relevant assays. So that's the platform. It's enabled us across all of our programs actually for genetic target validation through to molecular mode of actions, biomarker discovery, and drug testing. And it's also allowed us actually now for the first time to do gene to function at scale. And I think it's always dangerous saying this, but I think this is one of the first examples sort of ever of actually people being able to do high throughput pooled gene to function screens in human macrophages. And it's certainly the first example where we're aware of where IPS derived macrophages are being used that have all those benefits of really looking a lot more like macrophages you would find in a patient. So we've now shown we can do this at scale. We can do it at thousands of genes at a time in the human macrophages. And we've successfully run our first screen that's yielded some really interesting hits for us against a sort of therapeutic endpoint we're really interested in. So we're in the course of validating that now. So in the last couple of minutes, I'm just going to give you some highlights from the lead program, and then I'm going to introduce the team, which is sort of last but definitely not least. So our lead program is a LILRB1/2 antibody program. This is probably one of the hottest targets in the macrophage space right now. We're not the only people working on this target. There's a lot of competition out there. And one of the reasons for that is it's super well validated, and it has super robust biology, and it also has a multi-hit therapeutic hypothesis. So by targeting both LILRB1 and LILRB2, we're able to do multiple things at the same time. So we can essentially drive pro-inflammatory cytokine release from those macrophages to activate T cells and NK cells in the tumor microenvironment. It's really those cells that then can go on to kill the tumor cells. But also by blocking LILRB1 at the same time, we can also induce macrophage phagocytosis and also directly target and activate T cells and NK cells. So we bring all three biologies together in one antibody actually. And we're only one of 2 companies that we're aware of that has this sort of dual mode of action. So just by doing that, we've got ourselves into a relatively differentiated space, but we've actually differentiated far more than that, which has really been enabled by the platform. And we've brought together a really unique combination of sort of antibody features or assets to achieve what we think is going to be a superior clinical benefit. So the target's really, as I said, it's a really interesting target. There's some really encouraging early clinical data now from phase 1 from selective approaches that are targeting one part of that axis. But we've also actually identified an antibody that has a completely new mode of action. So this is something different to the dual targeting. That's a different aspect. We've actually identified an antibody that has a completely new mode of action in terms of how it targets these receptors. And we've also identified a potential blood-based biomarker for patient stratification, which is, as I'm sure you're aware, a major challenge in the immuno-oncology space. So we're bringing a really well-validated target together with a new mode of action and a way for stratifying patients. And here's an example of why that mode of action matters. So it actually leads to a completely new quality of antibody activity. So on the left, what you can see there is some data from a couple of slides ago, actually, where I was showing you data that the macrophage state predicted T cell expansion post PD-1 therapy. And one of the reasons people are really interested in T cell expansion is it's a surrogate endpoint for response to PD-1, PD-L1 therapies in the clinic. So patients that respond to those therapies tend to see much higher levels of T cell expansion. And you can model that preclinically. So what you do is you take macrophages, you mix them with T cells, and those macrophages from the same individual, derived from the same individual, and those macrophages are really immunosuppressive. Then you stimulate the T cells, and basically what you see is there's no real activity because the macrophages is essentially switching off that response. And you can use that system to test your antibodies or even your small molecules, whatever you want to do. And the endpoint that you're really interested in is in T cell expansion. That's the same endpoint that is used in the clinic in patients that predicts response. So it's the same endpoint we use preclinically in this in vitro co-culture system. And what you can see here on the right is kind of a head-to-head benchmark of our candidate antibody here on the right with some of the other antibodies that are currently working in this space. This is a dual antibody here, the NGM antibody, and there's others there as well that you can see listed. But hopefully what you can appreciate is that our antibody is driving much greater levels of T-cell expansion in the system. And we think if we see that in patients, then that's going to translate to much more profound clinical responses. And this has really been revealed by the technology toolbox that we have, and through this new molecular mode of action that has allowed us to identify. So very briefly, because I'm aware I'm kind of at time, more or less. So we're founded by Geoffrey Pollard and Luca Cassetta 3 years ago, really world-class founders in the macrophage space. So our CSO is Carola Rees, who formerly led Roche's innate immunity department and discovered the CSF1 antibody enmetuzumab. And Chris Doff-Witcher is our VP of drug discovery and was formerly at Kymab and Oceanics MedImmune. And I'm delighted as well also that Luca Cassetta, one of our co-founders, actually joined us as our VP of immunology 2 years ago. We're based in Edinburgh and Cambridge. We're hiring. We're about 16 people right now. We've got a great team. So if there's anyone who's on the call now or listens subsequently on the app, and you want to be part of the mission we have at Macomics to really develop transformational therapies for cancer patients, give us a call. We're currently hiring great scientists. Okay, that's it. And I'd just like to thank our funders and sort of partners for the company.
Thank you. Excellent. Thank you so much, Steve.
Welcome.
No questions so far. We are a little bit over time, but just very quickly. So is there a plan for your therapy-based approach to be combined with other cancer therapies, sort of chemo, radio? Is that how it works? Or?
Yeah, almost all patients, certainly in earlier lines of cancer therapy, receive combination therapy currently in the clinic. That's very common. And that's certainly, I think, a trend that's only going to increase. I think what's important, though, from a sort of a financing of venture capital, but also a sort of an exit, kind of from an exit perspective, is a monotherapy rationale.
Right.
So I think, clearly what you aim to do ultimately in your early stage trials and your phase 1 trial is demonstrate monotherapy activity, which gives you a strong base for combination activity. But if you have an absence of monotherapy activity and your only therapeutic rationale is in combination, that's much harder actually to generate early clinical trial data that you can kind of believe in, and that will translate into later clinical studies with a degree of confidence. And it's also harder to fund those sorts of therapies, frankly, and to ultimately exit from an investment perspective. So across our whole portfolio, we only work on targets where we think we have a strong monotherapy rationale.
Very interesting insights. Thank you. That's really valuable information. Thank you very much again, Steve. Thank you.
No, you're welcome.
And now a swift handover to Guy Hill. Thank you for sharing your screen already, founder of Daset Labs. So over to you, Guy.
Well, first of all, just say thank you very much for having me. It's a pleasure to be here. And if at any point, Alicia, you can't hear me, just wave a hand and I will, I'll put my earphones in, please.
No problem.
Just kick things off. So Daset Labs, we are all about empowering biotech, medtech to build a strong data asset part of their business. And probably a few people are going to say, what's a data asset? And a data asset is really just a collection of all the often biomedical data, but other data the company may wish to assemble, which adds value overall to what it's trying to achieve. And that's very simple. I think maybe it's kind of preaching to the choir, but in today's world, I think everybody is being asked how data is impacting their business, how it's being used as, for advantage. And so, maybe these words will start to come round, and you might even start to ask about your data asset strategy. It's something that we feel is very important. So just here, a few examples of what we're doing as a business before I kind of go and unpack some of these things. So as a company, we're very new, and a lot of this has been incubated in a previous company I've been involved in, Trajectory. And over the last year, these are sort of projects we've been involved in. And so right at the start, data asset strategy. So one example there for a large Japanese medtech, we got involved when they were struggling to develop a health monitoring app using a large amount of clinical data from annual health checkups. Really challenging space. but they were stuck. What we were able to do is to really unpick that for them, what that really meant, and the sort of forms of data they needed. I mean, really important things, the distinction between research data, commercial data, how the interplay between those. And that then leads to, in a sense, a sourcing strategy. What data does the product company need to bring in-house to move that forward? And how's that actually going to be used in practice? So things that we're able to help them with. And it's another sort of flavor of work that we do and been involved in the last year is essentially data scouting. What's out there, and how can it be, how accessible is it? And we sort of bring maybe three kind of aspects to this. One is just our experience of working around biomedical data for a long time. And another is some tools that we've built to help navigate and search for data in locations across the world. And that often involves mapping out a lot of the actual features or metadata of those data sets and helping to navigate them. And the third thing over the time, we've been building up our own kind of, I would call them just light databases of things that we can help get quick answers to. So if you're stuck trying to figure out what is out there in the world, then do tap us on the shoulder and ask us. We'd be happy to try and kind of help shortcut some of this for you. And that often leads into another area of work that we're involved in, which is data access and brokering access. Now, some of this is fairly straightforward if it's repositories where there's a real kind of push to kind of make the world aware of what's there and help get it out to be used. But if it's within a company, it's a much more complicated piece, this. And so many people have heard of kind of big company data sets such as Murative. I mean, there'll be others coming along, there's many others out there, but really complicated to actually partner with and to get access, and whether you're actually going to even really wish to bring that kind of scale of data into a company. Those are the sort of things that we've been involved with. And I want to just this all sounds very waffly in consulting world and kind of offering advice, but actually a lot of this is really practical, hands-on. And that's the fourth piece here of data cleaning, harmonization, big challenges there that we were involved in. So we are very hands-on. We are not just kind of advising on strategy and helping people get the data into the company. We're setting up infrastructure, we're cleaning data, and we're actually really pushing to be leaders in harmonizing these complex biomedical data sets as well. So we've just been awarded an Innovate grant in that space of harmonizing biomedical data. So I'm just going to push on, right? I've used up most of my time on one slide. So, I mean, what is a data asset? I mean, I guess we probably covered that a bit. It's something that adds value to the business overall, and you can think of that in a few different ways. But I'm just going to kind of just put a biotech related kind of focus on this about why do you need a data asset at the heart of the business? What's good about this? Well, I mean, just a key thing for biotech often there's a real medical thematic focus. I mean, it's very common that a company will have a kind of Alzheimer's disease and epilepsy. I don't know, you know, pancreatic cancer, but you can give any number. But there's a real medical focus within the company, and so there might be several products at various stages in development, clinical, preclinical. But overall question will be to the company, like, you're real leaders in this space, but are you leaders in owning the data asset that underpins all that as well, and the things that can come from that? So that is everything from kind of quite commercial things like market intelligence, designing kind of optimal approval pathways. There's so much happening in approval pathways, but you really do need the data at the heart of that to know really what's possible. And as a leader in that space, you're really going to be looking to command, be authoritative in that space as well. And that's particularly maybe for medtech as well at the moment as well, but in biotech as well. And the FDA, you see, is really opening up to the uses of data in the approval pathway. Come up for us in one project, difference between actually different sub-indications. I mean, a lot of this is, the world is moving so fast, especially in many of these disease areas where things look like they're clustered together. Are you actually even are you a leader in understanding the different indications, the interplay between those, and where the lines are drawn? I mean, obviously, discovery and research data, I think that comes. I think it's pretty. Those are things that we kind of hear a lot. I think everyone would kind of nod. Makes sense. And finally, things, just another thing, just at the point of actually the company going to sale as well. You think a few years down the line, there's going to be a competitive space. Do we have a transferable asset that goes alongside what we're doing as well that supports it? And that's the additional value. And are people just expecting you to be leaders as a data-driven company as well? I'm just going to hit you some challenges. Lots of strategic traps here. You can think about people get over-focused on the data they've got in their hands. Like, we generate all this data, this is really useful, but it might not be overall the asset, which is going to be the asset of collection of data that you need to be building together. So there's lots and lots of traps there. The finding and awareness of what's out there, that to me is still a piece that I think is creating a lot of needs for people. We get a lot of questions from people about what's out there and how can we find it. Legal compliance, that comes up all the time. I mean, at a very simple level, understanding the difference between research data, commercial data, but at the heart of it is understanding the terms of use, what's in the MTA, what that means, what you can't do with it. But also, think of it in a broader way as well. As many research bioresources, if you actually look at the terms of use, as long as that work is published, that can be used in a commercial setting. It's not precluding you to use the data in a commercial setting, but you must publish it, and there's often a requirement. But that can be, there's a lot of public good in most companies. I think on Nucleus, I think you say, are doing public good. They're about developing some making medicine better, trying to help patients. Lots of other things we can talk about. Competition, lots of other people trying to snap up data and getting exclusive licenses. It's a big problem. Okay. I'm just, in the interest of, I had some sort of taster slides, but I think I'm just going to skip very quickly over these. Just as if you're a biotech and you don't have there's no discussion inside the company of, what is the data strategy and what's the data asset strategy. My personal advice would be, please have some conversations, even if it's a very, limited conversation of just, okay, let's just have a sit down, spend an hour, and just remind ourselves of the big picture of what we're trying to achieve, where we're going with this, what data is needed to get there, what's an optimal data set to do that, and how much have we got internally of that, how much we're going to have to bring in-house, bring inside the company from outside, and where that might come from. Just a very, very simple framework there, just to have some good conversations. I mean, obviously, it can be much more complicated. Medtech companies, I think, especially where data is at the heart, going to be a much more complicated conversation. As an example here, I'm not going to go into too much detail. But if you're generating lots of data, and you're building a very, very large biomedical data asset, then lots of questions come along around mission, what's aligned, what's outside, how do you actually exploit that, and you actually get real value from that, and examples. Really for biotech companies, this is just example results. Everyone's going to know about it, but this is kind of just to poke everyone, saying everyone should use this, and it's the UK Biobank. I mean, it's the first one. Britain's an absolute world leader in this. Created a fantastic resource. Lots of companies accessing it. Lots from US, China. Not everyone in the UK, but we see approvals, but it's good. But just a reminder for everybody that the amazing resource that's there, and please just go and please use it. So it may only be 500,000 participants. Thank you for them for giving up their time, but it's very, very well curated against other things. And there is a longitudinal component as well, as they go back and resurvey. And of course, there's the genomic data, and just coming online at the moment, a lot of proteomic data for Olink as well. So that's coming on this year. And that's been paid for by companies like Janssen, etc. And then they've had kind of her first access to it, but that's now available for everybody. And so a great resource. If you're interested in doing that, the timeline is getting shorter. There's a good answer. So applications are getting quicker. But you really probably want to factor on, maybe 20 weeks, maybe maximum, at that kind of level. It would change during the year, but if you think about all the steps you've got to go through, and this, you might be able to go faster depending on how quickly you can draft applications and get through the hoops and things, get registered, and review the documents and do the bits you've got to do inside the company. But it takes a bit of time, but you're going to be thinking about, having access to that maybe for 3 years to start with for your project. So again, this is, thinking bigger picture here about the value you can get. And you can compare it against other things, other simple example sets. There are things which you can get access to almost immediately. But the power of the UK Biobank is incredible. So, anyway, but when you put things out there, you will also publish a short summary of the sort of things you're involved in, and so you can, it's useful to show some data. I don't know if we like to do that, but you can see sort of the trends of things here. Interestingly, much more recently, much more use the word patient. So, the people registering on UK Biobank undertaking studies have gone more from sort of looking at participants, looking more clinical and patient focused there. General harmonizing biomedical data. So this is an Innovate project we've just been awarded, where we're looking to conduct work to develop a prototype for harmonizing some particularly well-used biomedical data sources and the many things that fall out from that. And I think when anybody who's been involved in using biomedical data in detail will know the challenges are so multifaceted, and some of them unsolvable, some of them very, very simple to solve. But in the end of the day, if not really thinking at that level about developing a very high quality data asset at the end, then it falls apart. So this is really critical piece, the harmonization piece, really critical to developing a strong data asset. Well, that's us. But I mean, just to say, Alicia, thank you very much for the time and the opportunity to kind of talk. And I think there's a lot of things we've probably skipped out on. We're a small group of 4 people at the moment. We're about 2 months old, registered, and based at we work on Station Road. So if anybody, thinks some of this could be of interest, or struggling to find the right data for their projects, I don't know, come and have a chat. We'd be welcome to have a chat and have a coffee with anyone.
Excellent. Thank you very much indeed, Guy. In the interest of time, I am going to move Sophie on, but just a comment from me. It was really interesting to see actually, so your point around sort of, data being utilized to sort of help with companies' operations and streamline, that really does tie into, or the value of data, a bit to what sort of Frederick was saying at the end of his last presentation. That data is the next enabling step, isn't it? So having a data strategy, data management strategy is really a key consideration for any biotech, which is really what you've helped to
agree more. Beautiful.
help to showcase.
No, that was perfect. That was much more eloquent. Yeah.
So with that, I'm going to invite Christopher Mosdale. Oh, there he is. Sorry, to the virtual stage. Chris, are you okay to share your own?
I should be. 2 seconds. Let me see if I can get it to work. One participant can share at a time, so I don't think I've got permission yet. Ah, there we go. Where are we? How are we doing time-wise? So I can try and keep this one relatively quick.
You're fine. We've got some margin to go over a little bit. So no, please go ahead. Yeah.
Oh, don't worry, I have a reputation. We can run this one through relatively quickly. There's about 4 coffees in me at the moment, so this will fly by, I promise. So my company is Matrix Bio, and in the theme of data, what we are using is a tech bio term, full stack cell culture company. So that is looking at cell culture from a data-driven perspective, and we're trying to build a platform that will effectively just radically increase the speed and scale of 3D cell culture development and discovery, which is something that is all too slow at the moment. So as we know, we're talking to mostly biologists in the room, so I'll keep this one short. Biology is helping us grow everything, but mostly what we focus on is the DNA. There's so many technologies and tools out there to optimize and improve the DNA, the expression profile, CRISPR kits, you name it. But we're still quite archaic a lot of the time when it actually comes to the environment and the signals that cells produce. And cells are very, very sensitive to the things that are around them, especially as you get more complex and drift towards mammalian cells. So What we're trying to do is help biologists design and improve these environments using easy-to-use tools and a platform to do this within. So again, biology is taking over everything. We talk about pharma. We're in a Babraham-based campus at the moment, so of course, pharma is a big top conversation. But chemicals and materials, so many things are now becoming biologically derived or biologically processed. And of course, food and agriculture is seeing another renaissance in terms of its use of cells and biotechnology to improve that. So cell scaffolds are almost everywhere. What do you grow a cell in? If it's not just liquid or 2D media, it will be a cell scaffold of some description. And they're used across biotech, and they're extremely powerful tools which allow us to signal and control cells. They will guide fate, they will guide expression, they'll tell the cell where it is in relation to anything else. But right now, they're hard to design and they're hard to engineer. That's because 95% of designs fail with the way we do things. We're still very iterative, we're still very cyclical, and it costs a lot of money for something that should be as easy as ordering Matrigel. So in the ideal world, this would just happen. But in reality, it's expensive and we don't understand the 1,001 interactions in between, which is why 70% of the market is still relying on animal-derived products, which are essentially just a blend of 1,000 different growth factors, hormones, signaling peptides. And it just about works, just about all the time, but that costs 10 million mice a year, and it's just not really suitable for the new world we're moving into in terms of organoids or anything else more complicated. And we believe that by combining high throughput technologies, data-driven design loops, and prediction technologies, you can shorten the process, reduce the costs, and ultimately improve the outcomes. As I just said, that's the whole plan here. It's high throughput hydrogel engineering coupled with screening, which is an established technology. It's putting that into a design loop with computational tools. And this is creating new tools and systems to do something that we've never quite done before. So what we're doing is we're using generative learning at the moment to engineer beta gels and scaffolds. So customers can have a design studio, which has prediction software, allowing them to order discovery kits to get an idea of what might and might not work. And of course, we can do off-the-shelf or customizable scaffolds in between. Some of these will be royalties and licensing as we evolve into different workflows. Maybe there'll be drug discovery workflows, disease models that will be discovered and they might be worth pursuing. But generally, this is a data-driven company. And of course, that's why we can also do experiments and provide access to experts for different issues that haven't quite been understood yet. These are some of the tools we're building at the moment through the Babraham Accelerator. So designing the 3D scaffold, which should you be using? Should it be a self-assembling peptide? Should it be methacrylated gelatin? There's no real way to do this right now, apart from just looking in the history books and seeing what's worked before. And then we're going to try and integrate manufacturing so we can generate protocols rapidly. And most interestingly, factor optimization. I mean, it's the adhesion peptides, it's the growth peptides. These are the things that fundamentally change what a cell understands where it grows. A little bit about me. So my background is biology, long ago now, Thermo Fisher Scientific, and I've been involved in various spin-outs before this one. But most interestingly and most relevantly, I took a detour out into consumer for a while, and that was a D2C technology. And in that, we taught ourselves machine vision. And it was a machine learning enabled manufacturing process we built out in the center of London, which seemed madness at the time, but it's become more relevant. Our tech advisors are someone that helped us design our very first synthetic peptide. He's from the University of Cambridge, now running his own lab, Dr. Will Shu. We have someone who was acquired from Deliveroo, and all he does is he builds B2C web applications and platforms with that consumer focus in mind, which biology is still sometimes learning how to do. And possibly the most excitingly, we have someone who commercialized a biogel product in the past. That company wasn't successful, like many of these companies are. And she's very experienced in the world of pharma, but also has seen some of the trials which have affected biogel companies as they've tried to change this industry in the past. And I have a fractional CTO, Max. He is at the Sanger Institute. But before that was self-taught in the entire world of ML and DevOps. And that was in Canary Wharf, all around fintech, before having a moment of truth and deciding to come join the Sanger and teaching himself bioinformatics. So he's helping us build out this platform. The market is big. I won't bore you with the details, but it's a $1.5 billion cell market. $1 billion of this is in the organoids and iPSCs, which are used every day in pharma. We're targeting the 8 most common stem cell lines, seeing how we can optimize them in a $150 million a year market. This market is going to grow. We're already seeing it starting to grow, but we need to be building flywheels in the back end that will make this happen. So there's 3 flywheels that we see as important: data, which is driven by faster and cheaper with fewer experiments, generating more data overall. Sharing, having a platform where you can share and collaborate with different designs. And of course, cost. So what we want to try and do is make this cheaper and cheaper over time. So that's going to be one of our major KPIs. None of these gels are cheap, but fundamentally, they should become as cheap as possible to make more experiments happen. Oh, one skip. So to get into the Babraham Accelerator, we had a collaboration with the University of Edinburgh and their AI team at the Data Lab, establishing a very light deep learning engine using the 20,000 data points from the academic literature, which has its problems. Academic literature never publishes its negatives. So it's a very optimistic database, which we're now validating in the lab. And we licensed some proprietary gel technology from the University of Strathclyde. And we did some customer validation to actually make sure that this was something the market needed before entering the Babraham Accelerator. Through the Babraham, we worked out that there were over 2,000 cell lines in AstraZeneca still using Matrigel standard. And this is becoming harder and harder to do. So we know it's worthwhile as a company to start tackling PharmaFuss and helping them support their move away from anything animal derived, which is a massive regulatory burden at the moment. And right now we've calculated that that could be for a single drug screen, you're saving up to 120,000 every cell line you can replace with an optimized gel product. What we're doing right now is we're validating these tools across 3 projects, and we have 2 pilot customers using our tools. So this is an edge-funded process with the Medicines Discovery Catapult, trying our machine learning tool on oncology cell lines, as directed by AstraZeneca, a CPI-supported 100K collaboration, which is to develop that manufacturing integration. And we're releasing discovery kits into the market so that people can tell us whether our predictions are working or not. And that's mostly with SMEs and academic customers. And we are raising SEIS round at the moment. So that's part of the reasons why we're pitching here today. But I will stop there in the interest of time.
Thank you very much indeed, Chris. Just a comment again from me. I love the way the presentations today have highlighted the teams behind the business. It really does indicate, you know, how valuable the people element is to kind of creating these innovations. So yeah, that's been really fascinating to see that, and alluding to some of the future directions as well, which is quite exciting. There was only one comment in the chat box, which actually relates back to Steve's presentation. Marshall, I don't know if you wanted to unmute to ask that or not, or I can pick that up for you.
Hi, Alicia. Yes, this is Marshall. Yeah. Yeah, I'm asked the question to Steve, and it's a great, you know, technology platform and pipeline of projects. And I want to know is what stage is reached. Steve mentioned the clinical data, whether it's a clinical proof of concept is reached or not. And also, second question is whether Steve can consider, you know, a regional license deal. Yeah.
Yeah. No. Thanks for the questions. I think on your first question, I think there's an increased expectation and sort of that immuno-oncology agents will be immuno-oncology agents of the future that should be progressing into clinical trials really should, if possible, have a monotherapy hypothesis. I think if you look across the data sets over the last sort of 10 years, there has been some huge successes, CTLA-4, PD-1, PD-L1s, more recently LAG-3, and the kind of ongoing saga around TIGIT and whether or not that ultimately is going to be successful. But I think there's also been many failures, and in particular the failure of early stage combination trial data to predict later stage combination efficacy. Because obviously the combination agent, the standard care agent, can confound that data set in the early stage trial population. It's very hard to read those trial data sets. Patients are very sick, they've received multiple lines of therapy, they're typically not large patient numbers, and they're typically not biomarker led either. So from a Macomics sort of strategic perspective, we've got a really high bar for the targets that we work on. So we only work on targets that where we think there is a monotherapy hypothesis, and often that involves trying to modulate multiple aspects of the immune system at the same time. So every project we work on actually kind of has some form of multi-hit hypothesis as part of that. The other aspect is that we we really challenge, and I would encourage anyone in this space to do this, is we really challenge our sort of antibody or our molecule triaging early on in the program as hard as we possibly can. So we basically make those systems as immunosuppressive as possible to make them as much like a macrophage that you would find in a patient. And almost nobody does this, by the way. And if you look at a lot of the agents that are active in preclinical models, they're active in like what would be called kind of classical non-immunosuppressed macrophages. So we super immunosuppress the macrophages, and then many drugs, they don't work in that setting, and many of our own don't either. But it's a very high bar, and we think if you want to have monotherapy activity, you have to be able to show activity almost under the most hostile conditions possible. So we try and build that into the preclinical discovery, and then from a clinical perspective, for the lead program, we'll be going into clinical first in human trials as a monotherapy. We'll also have a dose expansion combination with standard of care that we think is relevant in the clinical context that we're aiming for. But we'll be certainly exploring the monotherapy and biomarker-led population. So that's the kind of strategy. And the reason for that actually is that when you're seeking financing from investors, what they want to see is what's your exit rationale. And actually, if what you're telling them is we're only going to have combination activity, and that's kind of what your data says, then what you're actually really asking them to do is to fund you at least to the end of phase 2 until it's realistic that you can kind of transact on that program. Whereas for monotherapy programs, the market is really, is really buoyant, and phase 1 kind of exit scenarios are realistic. So that's why it's important from an investment perspective. And I think on your question about regional deals, yes, I mean, we haven't done any regional deals so far, but yeah, absolutely, we consider them. And I'll put my I'll put my email in the chat, and please feel free to get in touch if you've got any other questions, if you want to have a discussion, or I'll send you a copy of the presentation as well. That's fine.
Yeah, that's great. Okay, thanks, Steve. Okay,
no worries.
Thanks, Marshall. Thank you, Steve. And thank you to all of the presenters and to all those who joined as well. So you can continue to use the conference app to keep discussions ongoing if anything comes to mind, as it often does post the session. Don't be shy. That's why the app is there. The recording will eventually be hosted as well for recap. Just a little bit of window of time in between that getting uploaded. So please bear with us. And then we will return at 1:30 today, so not too long from now, with an innovation workshop sponsored by CR UK on the questions you need to ask before translating your innovation to impact. So not to be missed. So we do hope you can join us for that. Thanks, everyone, and hope to see you in person as well on Thursday at the Cambridge building on Vabrum Research Campus. Thanks, everyone. Bye.
Okay, thank you.
Bye-bye.
Thank you.